Now that you have your prediction or a rough idea of your alternative or experimental hypothesis, you're ready to consider your statistical analysis. You'll need to know what type of inferential stats test you'll use. I'm going to use a t-test in our case, but which test you use is based on the type of data you gather, and we'll talk more about them later. You're also going to need to know your alpha value or level of significance, and you'll need a null hypothesis to properly interpret the statistical significance of your findings. Every lab experiment that will be analyzed with statistics should have a null hypothesis. So even though we read about them in lots of different papers, every paper should have them. A null hypothesis will seem weird at first, but it's not hard. Even though scientists tend to think in terms of our experimental or alternative hypotheses, we need null hypotheses in order to interpret our statistical results. This may be because early on statistics was used primarily for quality control. Is this batch good enough to meet our standards, or do we throw it out? It was all or nothing. No gray areas, and there are lots of gray areas in biology, so I think it makes the relationship between statistics and bio seem a little weird. It might have made a lot of biologists avoid statistics, and I think that's really sad because the tools that are offered by statistics are really cool, and I found them to be incredibly helpful now that I'm learning more about them. For my null hypothesis, I'm going to say that there will be no difference in the number of drops required to move tap water and blood plasma below a pH of 5, because I'm assuming there's no difference between blood plasma and water when it comes to their ability to buffer. In order to interpret my inferential test later, I have to assume that there is no difference between my control group, the tap water, and my experimental group, the blood plasma. I'll admit this whole null hypothesis thing is a little weird, since I am conducting the experiment because I think there'll be a difference. So we're going to need a threshold value for this quality control. How different do the numbers of drops for one group or the other really need to be in order for us to say there's a significant difference between the two groups? We call that difference our significance level, or alpha. The generally accepted alpha value is 0.05, so that's what we'll be using this year unless you give me a great reason why you're changing it. Alpha equals 0.05 is like 5% or 1 out of 20. What we're saying is that if we had two truly identical groups, like our null hypothesis states, and we run an experiment, say we run it 100 times, then we'd only see results meet the significance level simply by chance brought about by random sampling in 5 out of 100 of those experiments. So you could think of a coin. If my whole class flipped perfectly good and balanced coins 50 times, we'd see some pretty extreme ratios of heads to tails simply by chance. The alpha value helps us narrow down how different things need to be in order for us to consider them significant. So I want to show you a great XKCD comic called Significant. So here they are, they're investigating jelly beans causing acne, and they didn't find anything, no link. But then they think maybe it's only a certain color, and so they test purple and brown and pink and blue and teal. They're still finding nothing. And so they check salmon and red and turquoise and magenta and yellow and gray and tan and cyan and green. Wait a minute. Green shows something significant. And then they also check mauve and beige and lilac and black and peach and orange. Well, they just tested 20 different colors. And out of those 20 colors, one causes acne. I don't know. Have their misunderstanding of statistics led to their false conclusions? They did come up with 20 experiments, and only one color caused acne. This comic highlights exactly why we run an experiment one time, and we don't just keep gathering data until our statistical tests tell us we've got a significant finding. Technically, you could have no difference between your groups and you'd still find something significant 1 out of 20 times. It's a false positive or a type 1 error. If you set your alpha lower, this will happen less often. For example, alpha equals 0.01 will only give a false positive 1 out of 100 times. You may have noticed that the comic had p-values. P-values are compared to your significance level. So if you have a p-value of 0.03 and your alpha is 0.05, you can say your results are statistically significant. Use statistical tests to generate p-values, and this 0.03 tells you that if your null hypothesis were true and your two groups were identical, you'd only observe such vast differences between the groups in 3 out of 100 experiments. That's pretty rare. It's even more rare than our alpha value suggests. So we say our results are significant when p is less than alpha. Does that mean there's a real difference? No, but we accept the possibility of that type 1 error rather than throw out findings that might be interesting. If you're confused, don't worry. This stuff confused me for over a decade, but then I read a brilliant book by a brilliant statistician and it exposed me to this courtroom metaphor. So here's the metaphor. In a courtroom, people are innocent until proven guilty. In an experiment, two groups are the same, the null, until they are different. In a courtroom, evidence is required to show guilt. In an experiment, evidence is required to show differences. In the courtroom, jurors can only reach a guilty verdict if there is enough evidence. They may feel like the person is guilty, but without enough evidence, they must say not guilty. Guilty people can walk free, and innocent people can be convicted. In an experiment, scientists can only say their results are statistically significant if there is enough evidence. They may feel like their work is important and exciting, but sometimes interesting results are deemed not significant, and sometimes really uninteresting and inauthentic differences are claimed as significant findings. That's your type 1 and type 2 forms of error. In a courtroom, you are guilty or not guilty. We don't claim anyone is innocent, even though that's the presumption we start with. In an experiment, we reject or fail to reject the null hypothesis. We don't ever accept the null, even though that's kind of the presumption we're starting with. Rejecting the null means finding enough evidence to show there's a difference, and failing to reject means you did not find enough evidence. Guilty is like rejecting the null. It's why you went to court or why you started your experiment. Not guilty is like failing to reject. It doesn't mean the person's innocent or that your findings aren't interesting, but it does mean that your trial or experiment failed to show that with enough evidence. In a courtroom, there's no double jeopardy. You can't keep trying someone for the same crime. And in an experiment, there is no continuing to accumulate more data until you happen to show a significant difference. If you see scientists doing this, stop them. It's very bad practice and it makes us all look bad. You can convict the same person of a new crime, or you can redesign your experiment to try and answer the question in a new and novel and more powerful way. So I hope this helps you kind of clear up why the null is valuable and how it's related to p-values. We're going to keep practicing with this on your experimental design diagrams and your proposals for your IBET projects. You're going to write null hypotheses. So I want you to remember a last couple of details. Remember that null hypotheses are related to p-values, and p-values are compared with alpha so that we can decide to reject or fail to reject the null. That's it for now. See you back in class.